And welcome everyone. We're here at King's College London in the Centre for Stem Cells and Regenerative Medicine today, and we're going to discuss challenges and opportunities in regenerative medicine. My name is Fiona Watt. I'm Director of the Centre for Stem Cells and Regenerative Medicine, and I'm also Director of the UK Regenerative Medicine platform on immunomodulation of stem cells. I'm going to be the chairman for today's panel discussion, and I'm going to introduce now the other members of the panel. First up, we have Francesco Dasi, who's Professor of Regenerative and Hematological Medicine and the lead for cellular therapies at King's Health Partners. Then we have John Mayer, who's a clinical senior lecturer and honorary consultant in immunology. And finally, Graham Lord, who is Professor of Medicine and Director of the NIHR Biomedical Research Centre at Guy's and St Thomas's Hospital. So, as we know, the goal of regenerative medicine is to replace damaged or diseased tissue and find ways of treating diseases that are currently have a very poor prognosis. At present, two different types of approaches appear to be feasible. One is to stimulate the body's own cells to repair the tissue, and the other is to transplant cells into the body. And both of these approaches are classed as cell therapies. We're here to discuss the challenges and opportunities of cell therapies, and we're going to cover topics such as how cell therapy research is regulated, the role of the commercial sector, and how this research impacts on clinical trials. So let's begin by looking first of all at compliance. Francesco, what is the regulatory landscape in the UK, and is it supportive of cell therapies?
So this is a key question because we all were involved in cellular therapies require a lot of guidance. The regulatory hurdles are massive, and the main bottleneck for us to finally develop what we want into the clinic. So in the UK we got, I think, a fantastic regulatory agency which is called Medicinal Healthcare Products, or MHRA, which is meant to not only regulate but also advise in this. They, if I may summarize briefly what they do, I think that they are extremely important, also for personal experience, in facilitating the identification and the navigation of the most appropriate path to go through. And to give you some examples of what they're doing and have done, for example, they have an innovation office through which companies or academics who want to develop a product, they can contact them and be routed towards their specific need, whether it is a preclinical or clinical investigation or a process validation. They have also implemented a system for a fast track to access potential medicinal product, by which a company or an academic can make a particular medicine available even before it is licensed or has terminated the proper clinical trial. This, of course, has a huge implication for patients because they can receive these treatments even before everything is set in stone for the market. They have implemented a fantastic collaboration with the MRC and the Catapult to develop different guidelines. That is, for example, has been translated in a cell therapy passport. So every cell has a sort of credential, a proper evaluation sheet. It's not mandatory yet, but it is a good presentation, a good facilitator for any agent, any company, any academic who wants to develop this. They're also advising on a supply issue. This is particularly important again in the developmental phase. Just to give you the example, we have been working and developed a national mesenchymal stem cell program, and we are supplying at the moment cells based on a hospital exemption rule and the it is according to the specials. So there is this possibility that according to their guidelines and their advice all through the process, we have been able to provide, I think, vital resources for patients.
Thank you. That's very interesting. So by and large, you would say that the UK landscape is, in terms of regulation, is supportive of the field. So, John, what do you see as the biggest regulatory or policy challenges in regenerative medicine, both in the UK and internationally?
So, I mean, I guess I would start by echoing what Francesco has just said. I completely agree that in the UK we are very privileged to have a very supportive regulatory authority for for first-in-man testing of innovative cell therapy approaches such as chimeric antigen receptor engineered T cells and other related cell therapies. So I think early stage is, as I say, we're very well supported. I would say the key regulatory hurdle to the development of cell therapies is at the later stage of development, really. And thinking really more globally, we have a number of issues that I think we need to deal with. So there are problems with inconsistencies between the approach that's taken in Europe to develop a cell therapy compared to the US. So, for example, the clinical trial application process that we go through here is quite different, actually, to the IND application that would be made in a US centre. And even beyond that, even within Europe, within the so-called common market, as we used to refer to it, there is in fact quite a bit of heterogeneity in terms of the way issues such as good manufacturing practice are implemented across Europe. And the kind of data that one needs in order to secure the approval of regulatory authorities varies quite a bit. And these kind of heterogeneities, I think, are very important because ultimately, in order for cell therapies to become a key element of medical practice, as I'm sure they will, we need to embrace the commercial hurdles that brings with it as you develop these new cell therapies through the developmental pipeline. And we need standardization, really.
So can I just ask you, to some extent, the heterogeneity between countries smacks of a little bit of sort of old-fashioned protectionism. But if one of the trials which is going on with iPS cells in Japan or the US, if it were to be spectacularly successful, do you feel that the UK patients would be able to benefit quickly, or do you think that everything would have to start again from scratch in the UK before a proven therapy would be available?
Yeah. I mean, I suspect in the face of highly compelling clinical data that it would be the former that would be the case. But it nonetheless remains, for me, that is the key hurdle that needs to be addressed. We need to have a level playing field. We need to understand what kind of data package we need to present to regulatory authorities in Japan versus Switzerland versus the UK versus the US in order to secure approval to market that cell therapy.
Thank you. So now let's look at the challenges and opportunities which are associated with the industry and the market. So Graham, what do you consider to be the role of the commercial sector?
So the role of the commercial sector is releasing investment, I think, at sort of the medium to late phase of clinical trials. And I think the important thing that we're learning in the UK right now is that these processes need to be run in parallel. So I think at the moment, as we've discussed, there's lots of very exciting early phase work and first in man, and I think the regulators understand that. I think in order that we haven't just shifted the roadblock 50 yards down the road, I think understanding how you move from a first in man, sort of 20 to 50 patient study to a 250 patient study is going to be absolutely key. That requires investment probably over and above what the public sector could afford out with a few, sort of flagship trials. And I suspect, again, the balance of risk to investment will be, how spectacularly successful these things are. So I think if there was a very successful RPS trial in Japan, I'm sure people would be falling over themselves to fund it in the UK. But that's likely to be the exception rather than the rule. So I think the commercial sector that actually are looking very excitedly at the cell therapy field at the moment, but have made sort of very modest investment compared to, sort of checkpoint regulators in cancer, are looking to have proven efficacy, proven scalability, and clearly the model of IP process protection is still developing as well, because that would be critical to successfully unlock sort of scalable commercial investment for the, if you like, the pivotal phase 2b/3 trial.
So the California Institute of Regenerative Medicine, or CIRM, has one approach to supporting the commercial sector is to provide loans for companies to partner with academics. I would open the question to all of you actually and say, what do you feel about the cell therapy catapult, which is funded by Innovate UK and is designed to foster commercialisation of these types of treatments?
Well, I think the catapult's a fantastic idea. It's a fantastic experiment. So I guess time will tell whether it's a successful model, but I think it's quite an innovative model that the UK is launching, both within sort of regenerative medicine and in other fields of innovation. So it is very exciting. And I think what you do see is industry coming to the table, and Catapult can help to broker those arrangements. And at the same time, I think what Catapult is doing is listen to industry to say, what is it we need to make this an investable proposition? So I think the large-scale manufacturing facilities, which they're going to be building in Stevenage over the next 18 to 24 months, is going to be absolutely critical to de-risk it for that commercial investment. So I think the Catapult model is very exciting.
So John, Graham mentioned immunotherapy for cancer, and that is an area that's very close to your heart. So could you say a bit about your own experience of going from proof of concept into a clinical trial, and possibly a bit about the interactions between industry and academia there?
Okay, so we have developed a number of chimeric antigen receptors over the years, one of which we have just taken into a clinical trial which began in the early part of this summer. It was kind of an interesting experience for me because the development of this program was initiated before CAR T-cell immunotherapy had attracted the kind of interest that is now certainly very prevalent and focused around B-cell targeted or CD19 targeted CAR T-cells. So it's kind of been interesting to watch the evolution of the field and our own experiences as we try to take some of these molecules into the clinic. What I would say is that our experience really suggests that the way forward is to engage with the regulators as early in the process as possible. So we made contact with the MHRA in order to seek advice from them in terms of the kind of data package that they would like to see in terms of efficacy of the approach, in terms of potential safety in relevant preclinical models, and also in terms of the quality of the manufacturing process that we could set in place in order to secure approval to go into phase 1. And I have to say, once again, echoing some of the comments that Francesco made earlier, that the regulators were absolutely fantastic in the kind of level of advice that they gave us, basically on an informal basis. But nonetheless, the steer that they gave us in enabling us to design particularly our safety studies was absolutely fundamental.
So can I ask, can you put a time frame on that? How long was it from your first approach to getting approval?
So it did take a long time, but I think a large part of the reason for the long time that it took was the fact that when we began, CAR T-cell immunotherapy was viewed, I think, as the kind of activity that people who had watched too many episodes of Star Trek as a child would be engaged in, and that this was a technology which would never make any kind of meaningful impact in the clinic. And we've gone from that situation. It's typical of things in life, really, where you go from one extreme to the other, where now we have an absolute frenzy of activity surrounding CAR T-cell immunotherapy based upon the efficacy data that have emerged from a number of US centres in particular surrounding B-cell or CD19 targeted CAR T-cells, where in a first-in-man setting we've got complete remission rates in patients with ALL of approaching 90% in more than one centre. absolutely amazing. Of course, the real question for people like me is how do we translate all of this into the area of greatest unmet need, which is solid tumours, because of course solid tumours account for 90% of the cancer and probably account for 95% of the unmet need.
So are you saying that if you were to approach the regulators now with a new indication for CAR therapies, the process of approval would be quicker?
I would say that if you were to set out with a good idea now from scratch, you could probably get into a clinical trial within 3 to 4 years with appropriate engagement with regulators, so that you design the efficacy and tox studies with their advice in mind. I would say 4 years is probably a realistic time frame.
And can I just ask Francesco and Graham, you've both got clinical trials either ongoing or approved. Would you say 3 to 4 years in your different indications? So Francesco first.
Yeah, my experience with mesenchymal stem cell for the treatment of immune-related disorders, and is probably a similar pathway. You still have to navigate where to get the money through, but it is certainly the time frame is exactly that.
And Graham, for you?
I think what you need to look at is whether you're going to take something sort of stock manufacturing process off the shelf, where sort of 2 to 3, maybe 4 years. So I think we have compressed that timeline. If it's relatively standard, I think what we're doing with our cell therapy program is bringing about new technology. So we're looking at
perhaps you could explain what your trial is going to be.
Yeah, so our first trial off the block, which hopefully will be starting in the next 12 months, is adoptive therapy with regulatory T cells for inflammatory bowel disease. So sort of moving away from cancer, if you like, the other side of the same coin, to an overactive immune system. And how can we take out syngeneic cells from a patient with Crohn's disease, for example, expand them in vitro, and then reinfuse them in order to suppress the aberrant immune activation in the gut, and then help the epithelial cells of the gut to repair. So in order to do that, because we were concerned about plasticity in vivo in an inflammatory environment, we were worried that the regulatory T cells might turn into effector cells and cause more damage than they sort of cured. We've gone down the line of developing GMP compliant flow sorting, so sort of FACS, fluorescent activated cell sorting, which is a new, if you like, manufacturing process that we put upstream of the expansion phase prior to adoptive cell therapy. And so that has expanded it a bit further in terms of the timelines. I think we've had some interesting early discussions so who is it that regulates it? So we talked about MHRA. Is it MHRA? Is it the HTA? And sometimes when you have very innovative technologies like that you're bringing to the field for the first time, I think the regulators sometimes struggle to see who's going to regulate it. But actually, as you say, early discussions can help with that. So I think the regulatory environment is also changing in an iterative fashion, and with discussions with investigators as they see the unmet medical need, and there's more investment in this field. So probably for the study that I'm running, it's been a little bit longer than that. But again, that's because there's been technological development. We also then have partnership with the manufacturer of the flow sorter, so we have to get back to the factory and re-engineer parts of the process, which again is very exciting. And I think one of the key points here is that once we've developed this for this one indication, it will be a platform for every single other indication that requires very, very pure cell products. And just to echo one of John's points earlier on about the heterogeneity of regulation, but also the heterogeneity of cells, the way that was solved in manufacturing small molecules or antibodies was ensuring purity and consistency. So I think these sort of platforms and these technological developments of the process are going to be vital alongside the science and the discovery of new targets to moving the field forward as quickly as possible.
I think you make a good point, because when we think about commercial partners, we often think about a partner who is going to fund a clinical trial or co-fund one. But in your case, the commercialization opportunities around equipment, devices, or reagents like antibodies are probably lesser risk for the company, and it's a real partnership, because success that you have in the clinic will lead to sale of more of the equipment or the reagents. And so you could see a cycle of mutual benefit with the commercial sector.
Absolutely. No, as you say, it's much less risky, because if a platform doesn't work in one type of indication, there's lots of other indications it can be used in, because it's about developing a process. And as you say, when you match the development of sort of hardware and then the software, as you might like to talk about, the reagents, if you match that together, these things are always going to be very expensive. They're not going to be the sort of thing that will fall massively at scale. There's a huge commercial opportunity there. And as you say, if we're successful, they're successful, and vice versa. So it's a proper partnership. It's been very exciting.
It's good. Now, the next topic that we're going to discuss is planning a clinical trial. You three are clinically qualified. I'm a PhD. And I know from reviewing many grant applications that there are many very successful basic scientists who will, in the final paragraph of their grant application, explain how they're going to cure heart disease or dementia or anything you would like. And basic scientists often find that if they genuinely want to progress their work to the clinic, they've probably left it a bit late because they have tried in a very scientific fashion to really nail the basics. And then they realize that if they had thought about the reality of going into the clinic, there are certain things they could have done earlier on. Is that a fair assessment?
I think it probably was. I think it's becoming less so. And I think one of the things that is a game changer is the investment by the National Institute of Health Research in sort of biomedical research centres. So I direct our biomedical research centre here, as you mentioned in the introduction. And what the BRC does is it brings together sort of basic scientists who are interested in translating, clinical scientists who are doing some of that early phase translation. So these questions are starting to be addressed earlier. I think what it also does, so it's not just a question of, if you like, sort of conceptual pathways and at what time do you jump into translation. I think it's realising that actually it's hard to do it within an individual lab. Even if you're very, very large and well-funded, and what you need is sort of critical platform infrastructure, which is here certainly what the BRC is invested in. So GMP manufacturing, immune monitoring, and all that clinical research infrastructure that you need that's cost tens of millions of pounds, that no matter how good a PI you are, it's always hard to raise that sort of money. I mean, we all write lots of grants, but to get that infrastructure with support from both a hospital and a university, I think is one of the things that makes this place unique. So then, basic scientists can see, this is how I can translate. They see it happening. So they start to have those thoughts and discussions earlier on. And the other wraparound you have that is, the regulatory understanding, as we've discussed, the ability to design a clinical trial. So what sort of clinical trial you want to do, first of all. And I think there's a lot of innovation going on right now around the world that's both commercial and academic about sort of early phase adaptive trial design. So we've been stuck with the sort of the 3+3 safety, yet safe, and that takes 5 years, and then we take another 5 years to do a bit of efficacy. So actually we're going in earlier to patients and looking for efficacy. So I think the whole model of phase 1A, 1B, 2A, 2B, phase 3 that's based around small molecule development and biologic development is not really fit for purpose. So I think there's innovation in all of this space, which is A, what makes it challenging, B, what makes it expensive, but C, what makes it very exciting to be involved in.
Thank you. Francesco, thinking about collaboration, I was wondering if you could comment on your experience of being a partner in one of the in the UK regenerative medicine platform, which, as we know, is an investment by the research councils of £25 million, really to build on existing funding to facilitate collaborations and identify and try to backfill areas of weakness. So what's your impression of UK R&D?
I think the MRC has been really an amazing innovative, produce innovative enterprise by building platforms across different disciplines. We know, our own experience, how we put together stem cell biologists and immunologists that probably a few years ago were almost impossible or didn't make any sense. And this has produced a fantastic network in which there are scientists involved in cell imaging, scientists involved in the behavior and the differentiation of stem cells, but at the same time, how can we modulate the patients, immunomodulate the patients to accept the different stem cells. So I think it has produced a fantastic opportunity, which probably even comes before the phase 1 clinical trials, because we have this with this necessity to get together, to have a really very good enterprise, to somehow echo and look in the perspective that a different perspective that Graham was mentioning. Commercials and companies have sometimes pressed, of course, to test new therapies. And I can talk about my own experience with mesenchymal stem cells. There have been, and also other stem cells, you know, Fiona. There have been some failures due to the fact that there was a rush into the clinical trial, without probably knowing better about science behind it. And this is at high risk of some time, you know, aborting a pathway that could be otherwise extremely promising. So I think the KRMP is a fantastic initiative to put together this underlying framework that can help us to go back and forth for whatever question we want to address in a clinical trial.
And looking beyond the UK to Europe, the Horizon 2020 funds quite a lot of activity in regenerative medicine. What are your experiences of those kind of networks? I think you're in the MSc one?
I got 2 trials. No, actually 1 trial and 1 other funding in that. I have to admit, our own UK evaluation of grants is much more rigorous, much more scientific, science-oriented, but if you like, a bit more conservative. I think that you know, the EU, if you like, has this advantage of being a bit brave, and they have spent a large amount of money on 2 calls exclusive clinical trial over the last year. So we'll see where this leads. Certainly they got quite a lot of resources, but still is a...
I mean, I think the EU networks are valuable on a number of levels. I think, as you say, they can be a bit more risky, whether that's because there's slightly less accountability for them. And I'm sure that's not the case. But I think what it does is it builds networks across Europe and understanding of the differences in the regulatory process, the difference in the manufacturing process. So my hope is that actually it can share best practice and raise the bar across Europe, because actually in terms of commercial investment, you know, whilst we're hopefully in part making the UK the best place to come and do regenerative medicine, which I genuinely think we are, if you're a global pharma company, you'll be looking at major areas, and it's actually the whole of Europe. We're actually too small a market if we've said we've only fixed it for the UK. But actually, if we fixed it for the whole of Europe, and then that's readily transplantable to the US, then all of a sudden you've got a very attractive commercial proposition, obviously if you can show efficacy and cost of goods.
So, John, I was going to say, I know for you it's been a long haul to get to your first clinical trial. Could you say something about the main routes for funding for phase 1 trials and beyond?
Okay, so I guess based upon my own experience, initially the funding situation was quite challenging, actually, particularly before CAR T cells achieved any kind of clinical efficacy, as I've referred to earlier. So often it came down actually to securing money from philanthropists, because it was very difficult to obtain funding from any other sources. The traditional bodies to whom one applies for funding for these kinds of studies certainly pays a lot of interest to the commercial angle of the therapy that you're trying to develop. And when it comes to cell therapies, that can be quite challenging, really, in terms of how does one envision to commercialize a therapy that is just pre-phase 1 testing. What's the intellectual property situation like? That can be very, very difficult, etc. So I found it quite challenging to obtain funding from the traditional sources starting out. With the emergence of CAR T-cell immunotherapy and the increasing interest in the pharmaceutical sector in this kind of approach, allowing for the complexities of the IP landscape, I found that the funding environment changed dramatically. So for phase 1 in the UK, Wellcome Trust, MRC and Cancer Research UK are all now very interested in this kind of activity.
And Francesco, what about beyond phase 1 proof of safety? What happens after that?
I think that is a big challenge in which, as Graham was mentioned before, we do need large investment. And this cannot be, of course, taken by the governmental agency and the funding which is currently available. So the largest phase 3, which would probably require hundreds of patients, do require big investment for the big companies. Not even sure, whether a small, medium enterprise or small biotech would be sufficiently able to afford that. So that is probably the biggest challenge. But for example, Europe is helping to some extent in this. All consortia have small and medium enterprises together, and this is an important aspect that can ignite this. I also think I want to make a point about the need within the cellular therapy of identifying probably a more restricted number of patients, because it is much more complex, not only regulatory but also scientific. It's not a drug, so we don't even know what happens after. Biomarkers are almost entirely unknown, and we do need that sort of support. So if we could also invest, as the UK is doing, in identifying some of the biomarkers which predict response to a particular cell, we could make our trials, even phase 3, much more effective without the need of thousands of patients.
So that brings me on to a question for Graham about patient recruitment. In Francesco's case, he is treating patients with graft versus host disease, which is life-threatening and acute. John is dealing with cancer patients who have exhausted all other avenues of therapy. But if you're approaching a young man who's got Crohn's disease, which can be managed, do you think it's going to be difficult to encourage someone like that to participate in a cell-based therapy trial?
I think it's a very good question, and I think, if you like, the bar to entry into a market's lower in cancer, for, as you say, for the obvious reasons. I think, and there's always this debate about, whether it's sort of pricing and market entry with autoimmunity versus cancer, which is and I think what people don't really realise, unless they're seeing people in clinic the whole time, is that there's a huge unmet need in autoimmunity. So our trial in Crohn's disease will be for patients with sort of moderate to severe disease who have failed standard therapies, and we have discussed this with our patient group prior to setting up the trial to do, if you like, a sort of a feasibility study, and patients are very, very keen to try a novel therapy for something that is a sort of chronic disease that affects their ability to hold down a job. These patients are often sort of quite young, in the sort of prime of their sort of economic and lives, in their sort of their career and wanting a family. So there's a huge need there. And a sort of huge market size as well. If you look at sort of TNF alpha blockade, which is the standard treatment at the moment for someone with Crohn's disease. So there's a significant number of patients. About 70% of patients don't either respond to TNF alpha blockade, or they don't have a maintenance of remission. So a very large patient group there. And actually, when you tell them that what we're going to do is take some blood, expand their own regulatory T cells and make them work and put them back in, it actually is. People say, well, that makes sense, because surely you've been doing much more ambitious things than that with kidney transplants or bone marrow transplants or islet transplants, which are now routine clinical care. So I think when it's explained in those terms, patients seem very enthusiastic to participate. And also, if you have a chronic long-term disease that, where you accrue disability and you need lots of surgery and lots of potentially toxic drugs, I think patients are only too keen to get involved.
So that brings me on to a question about infrastructure, and I'm thinking particularly about GMP laboratories for expanding cells. So the Cell Therapy Catapult is investing a lot of money in a large facility which will offer opportunities for people to expand their cells. And I could imagine that for some treatments, the ideal would be that you have a freezer or a liquid nitrogen tank, and instead of prescribing aspirin, you prescribe a vial of cells. But for other treatments, you're, as you've said, treating somebody with their own cells. So where do you think universities and medical schools should be positioning themselves when it comes to infrastructure?
So, I guess speaking from what our experience here, sort of seven or 8 years ago, we made the decision to invest in GMP manufacturing infrastructure as part of the Biomedical Research Centre, which again shows you how long it takes. So we're just starting to use it now to deliver our first in-man trials. I think university medical schools absolutely need to be in that first in-man space and have the ability to manufacture their cells. And critically, as Francesca was saying with the UK RMP, be informed by the basic science to say, actually, okay, this is our first one off the block, but actually this is our second phase, this is our third phase of regulatory T cells or CAR T cells. So there's continual process improvement that's informed by the science, the discovery of new targets, the discovery of new biology. I think that's absolutely core business. Where we're moving it to here is the ability to manufacture to intermediate scale. So we can manufacture for 20 to 30 patients now. We're very good at that. We've just done 3 first-in-the-world trials over the last 4 months. John's is one of those, where we've manufactured the cells here. Stevenage will be for phase 3 when you need 300, 400. Actually, there's a real need now, both for gene therapy and cellular therapy, for that intermediate scale manufacturing. And I think that's a perfect opportunity, as we're doing here, for public-private partnership between university, hospital, medical schools and SMEs, the charitable sector wanting to make investments for that intermediate scale manufacture, which is exactly what we're doing here to, if you like, move towards that large scale manufacture. So actually we can do the definitive, we can manufacture and perform, if you like, the definitive intermediate phase. And I think, as Francesca was alluding to, understanding where we can use adaptive trial methodology, I hope means that we can show efficacy that the regulators and critically the purchasers may start to, particularly with adaptive licensing, from smaller number of patients. If we're clever, rather than the standard, we need 5,000 placebo-controlled, the standard sort of placebo-controlled clinical trial that's been standard, which is more suitable for small molecules.
So, Francesco, I know that at King's College Hospital, you've been very heavily involved in setting up a GMP facility, and it's multiple facilities within the same core for gene therapy and cell therapy. So I know the experience has been painful in some ways, but could you give us some perspective on infrastructure?
As we all know, it's very painful. And I can tell you because... Well, exactly. I mean, just to give you an idea, I started at Imperial, when I was at Imperial, the mesenchymal stem cells, and that was about probably 8 to 9 years ago. I can't say it was easy, but it was easier. Redoing more or less the same thing just by moving to another facility is taking at least another year. We're not yet ready yet to reproduce the thing.
But is that because you have to start from scratch in terms of the regulators?
It probably is correct, because the standard operating procedure is not very different. You adapt whether there is a corridor or a wardrobe or something like that, but it's not that difficult. What is required, more quality control, and how we started in the beginning was probably relatively relaxed. When the new regulations have been taking place, this has, of course, made things much more complex. And the steps in quality control, as Graham and John know, are unfortunately endless. With what I'm not saying and not criticizing the MHRA, because they are fantastic in helping with this. But of course, the regulations have been adapted by the pharma with molecules, and in cellular therapies is much more complex compared to molecules. And that is, of course, another layer of complexity that is very difficult.
But I think if I can come in, I think this is slightly different between the sort of the US and the UK perhaps, is that I think what we're doing, and again, this is why I think the UK will be the best place for regenerative medicine and cell therapy, is we sort of front load our QC and our processes. Yeah. So actually, once you have something that works, it's a lot quicker to scale up. And I think that's part of the concept that the Catapult's helping with, that the MRC's invested in. And I think rather rapidly we've developed quite an impressive community of basic scientists, clinicians, hospitals and universities that want to invest in this, and funding agencies and commercial bodies that are interested in investing in it. So I think we've really got a critical mass now, and I think it is painful and it does take time and it's very expensive. But I think those are the sort of things that, again, if we can show efficacy, will pay off down the line, because if you like, we sort of front-loaded that investment.
I must say, I like your point about the partnership between basic scientists and clinicians, because I was very inspired by the BRC here to take some steps towards a cell therapy trial. And when we were funded to hire somebody to work on that, I thought, no postdoc is going to apply to my lab for something which is so practical. And I was absolutely delighted that the quality and the calibre of the applicants was fantastic. So I do think in terms of basic scientists, we're preaching to the converted. I think people want to do something useful, and they understand what we're trying to achieve. So that is I would say, is a major change in thinking about the potential of cell therapies.
If I may, actually it is a new avenue, and as you know, we have developed a new master in cellular therapies with a particular and very large module on the market initiative. So how to translate an idea from the bench directly into a viable business model. You know, also the PhD course that you have developed will fit very well with this notion, and to finally see what we do, how can it be, make a beneficial to the UK PLC.
Well, I think if I also, sorry, jump in here, I think it's also, we're talking about translation in one direction, which is great, if you like, for clinical impact. But actually, I think what we're also capturing here, and we need to do more of, is capture the reverse translation. So how do cells behave? Well, if you put them into a patient and you have the conceptual flexibility and the analytical infrastructure to look where they go, what they do, how they behave in the niche that they find in a patient, well, then there's a huge opportunity to, if you like, validate targets that we've discovered in vitro or in preclinical models, and also to discover new models that are specific to human disease. And I think understanding that, if you like, that reverse translation, to do really, really good basic science and reductionist science in patients with disease, I think is a relatively unexplored offshoot of this, but obviously we're well positioned to look at here.
I think we are. So we're going to wrap up in a few minutes, but we can't stop without asking each of you what you see as the future of cell-based therapies. So John, from your perspective, where are we going here? If you could think beyond the cancer sphere into regenerative medicine, are you optimistic, or do you think it's a flash in the pan?
I'm extremely optimistic. I mean, I guess I've based my whole career on the development of cell therapies, so it would be strange if I was to say anything other than that, really.
But you want to kill cells, and that's a bit different from treating diabetes with implanted beta cells.
Yes, I mean, I suppose I view cell therapies in general as living drugs, really. I think it is a new modality of treatment that is just about to enter the clinic on multiple fronts. And that is part of where the challenge lies, because there is a lack of familiarity at the kind of drug development, throughout the drug development process with this new therapy. But we have seen very, very compelling proof of concept of how cell therapies can achieve things that no other therapeutic modality can. So I'm extremely optimistic about the future. I think there will be challenges, particularly about cost of manufacturing cell products and about reimbursement. But the reason that I remain optimistic nonetheless is because I think technological advances in this field are happening so quickly that they will circumvent many of the obstacles that we see at the moment across the board.
And Francesca, I know you're a card-carrying haematologist. Are you ever going to be treating your patients with pluripotent stem cells? Or hopefully they're derivatives?
Well, that is a very interesting concept, because at the moment, for example, we could, consider ourselves happy with hematopoietic stem cells. But there are, of course, a number of challenges. And I would say in particular the cell dose. From our experience, we know that it would be better to have as many cells as we can. And this, as you know, this has been also experienced in other even failed clinical trials. So the somatic stem cells are not probably that good at being expanded. There is no good, I'm talking about stem cells, not T cells. Certainly the opportunity, IPS or something similar, is a great opportunity. Possibly more controllable. We don't know to what extent we can refine it, but it still is a great opportunity. The other aspect, whether this could be autologous or allogeneic, which is another big challenge, as you know, as a UK RMP, and our hub is specifically addressing these questions. But just to give you an idea, when Graham would never transplant would never think that an allogenic kidney would not be rejected. Now we start thinking that there are some cellular therapies that can still produce an effect without necessarily being identical between the donor and recipient. Even CAR T cells. We just injected the first patient in hematology today, actually. So it is a big challenge that could even challenge some of the big dogmas of immunologists.
So finally, to put Graham on the spot, because I know that you've transplanted the odd kidney, are you ever going to say you don't need a donor kidney because you know what, we've got some kidney organoids that you can have instead?
I think so. I think so, and I hope it will be within my, well, first of all, my working lifetime, because that would be very exciting, but hopefully within my physical lifetime. I think I'd probably say three things. One, is there's sort of a bit of a medical analogy, but people always talk about the ECG of development of new fields. So you get a bit of excitement to start off with. So that's your little P wave. So I apologise for the non-clinical trend. That's a little blip at the start of your ECG. Then you get this enormous, R wave of excitement where everyone's piling in to invest, and you see the CAR T cells and 90% cure, and people are doing deals all over the place, and that's where we are right now. There'll be a massive plunge in the next 3 or 4 years. I suspect the bubble of optimism will burst, and that's your big Q wave that comes right down. And then actually what it'll do, it'll settle back to baseline. There'll be what we call a T wave, which is actually the unmet need where these products will fit into. So I think we have to manage expectations of politicians who want short-term turnarounds and good news stories, and it all needs to be on the upward trajectory. So that's very important. I think the other concept we need to consider is combinatorial therapy. So by which I mean we may need to transplant cells in combination with administration of endogenous regulators of those cells, possibly in combination with checkpoint regulators in cancer, so with monoclonal antibodies. So I think that combination is going to be very interesting to explore. We'll be driven by the basic science, but actually critically from the reverse translation about what we learn about the PK and the PD of the cells that we do transplant. So the ability to track these cells is critical. And also the links with bioengineering. So what do you put these cells in? Is it, do you put them in a box? What membranes do you need? And how it fits into sort of nanotechnological targeting. And I guess the final thing is I think we're at the start of a really exciting wave of new science and medicine, very similar, I think, to when the first drugs were made, when the first monoclonal antibodies were discovered, and they all went through, particularly monoclonal antibodies, went through a massive dip, and everyone said they're never going to be used for treatment, and now they're the biggest blockbusters on the planet, treating huge unmet medical needs. So it's very exciting to be involved in just a very small part of that.
Well, thank you. That's all we've got time for today, but I'd like to thank all of you for a fantastic discussion, and I hope that our viewers have enjoyed it too.